Hi everybody, these are your notes on DNA replication. You may have to pause this video as we go so that you have time to write down the notes. We have talked about how DNA makes a copy of itself in protein synthesis. Remember that you need a copy of the DNA to go to the ribosome, and remember that whole process is done with a molecule called RNA polymerase. DNA replication is a little bit harder. There are more molecules involved here. So first, why do we need to do DNA replication in the first place? You need to do DNA replication anytime your body makes a new cell. That new cell has to have a copy of all the DNA. Remember that every cell in your body has exactly the same DNA in it, for the most part. And every time you make a new cell, then that means you have to put all the DNA in it. So anytime you grow, you get bigger, your bones get longer, your muscles get longer, you have to make more skin, your hair grows, your fingernails grow. You have to make more DNA so that those cells can divide, so that you can get more of those cells. Growth is just making more cells. Anytime you have to repair your cells, like if you get a cut or you break a bone, you also have to do DNA replication there to make a new cell. And then also, when you make sperm cells or egg cells, those cells have to have a copy of the DNA in it too. So reproduction. Okay, like I said, these steps are a little bit harder than making a RNA copy of DNA. With RNA polymerase could do everything. It could open the DNA, it could make the mRNA. There was nothing else that needed to happen. With this, though, it's more complicated. So first, you've got DNA helicase. DNA helicase is an enzyme. It has to come in, and it's what opens up the DNA. It breaks those hydrogen bonds, separates the A's, the T's, the G's, and the C's before you can even start to make a new copy of the DNA. This is called a replication fork here. So it kind of looks like, you've got a road, and then the road goes in two different directions. It's like a fork in the road. This is a replication fork. This is supposed to be the DNA helicase coming in and separating the hydrogen bonds. All right, step 2. Now we've got primase. Primase adds RNA primers. Yes, that's not a typo. It is actually RNA primers. I know we're making DNA. But DNA polymerase, which we'll get to on the next slide, needs a lot of help. So these RNA primers help to tell the DNA polymerase where to go, where to add DNA. It adds, primase adds the RNA primers, one to what's called the leading strand. This strand goes in the same direction as the DNA helicase does. So it just puts one on the end there. But then if you look at the other side, the green is the primers. So it adds a primer here, it adds another primer there, it would add another primer if it kept going. So it adds them in little pieces. Okay, so now we've got helicases come in, open up the DNA. We've got RNA primers in here telling the DNA polymerase where to go. So now the DNA polymerase, step 3, can come in and attach to those primers and finally start adding the complementary DNA bases. Those are in red here. So here on the leading strand, you've just got a single strand of DNA that's being put down. So these are, if they showed it in detail, this would be like the A's, T's, G's, and C's all pairing up with the right base. But then on the other side, DNA polymerase will come in here and add a little bit of DNA, and then it's going to come in here and add a little bit of DNA. And it works backwards. It does work in the other direction. So here you're making one single strand of DNA, and then here DNA polymerase has to do little segments of DNA. Step 3 continued. That strand that's following the helicase, this strand here, the leading strand, just gets made as one continuous piece of DNA. And then on the other side, this strand is called discontinuous. It's the lagging strand, and it's made discontinuously in little pieces. Those have a name. They're called Okazaki fragments. They're named after a Japanese researcher who discovered them, so he named them after himself, Okazaki fragments. This step just keeps continuing. You keep on having primase that's adding an RNA primer. You keep having DNA polymerase that adds DNA bases. Helicase moves down and opens up more DNA. And then primase comes in again. So that keeps on happening for all of your DNA. And remember that even though this seems complicated, it is simplified based on what actually happens in your body. So you can read about this and figure out there are steps that I'm leaving out. Step 4, there's actually a different DNA polymerase. We'll just go ahead and call it DNA polymerase though. That takes out the RNA primers because we are making DNA. We don't want RNA in there at the end, and it will replace that with DNA. And then there's a ligase, an enzyme here, ligase. It glues or it bonds those Okazaki fragments together because we don't want the DNA to be in pieces. So I think ligase links the Okazaki fragments together. So here's our picture again. Remember, DNA helicase opens up the DNA. You've got primase that adds RNA primers, one to the leading strand, the one that will be made in one continuous piece, and multiple RNA primers to the other side of it, the lagging strand of DNA. And then you have DNA polymerase that comes in and puts down DNA, continuous piece on the leading side, and then little Okazaki fragments on the lagging side. Then DNA polymerase will come in, technically a different one, but another DNA polymerase will come in and remove those RNA primers and trade them out for DNA. And then ligase comes in on this side and links together the Okazaki fragments. The product, so you get DNA, that was our goal, but this is sometimes hard for people to think about or realize, I guess, is that you end up with 2 double helixes. That should make sense. But each one of them has a new strand, so the blue, or an old strand, the blue would be my old strand, and a new strand. The red would be the new strand. So this is considered semi-conservative replication because it's semi, like half conserved, half the DNA is used again, like recycled, reused, and then half the DNA is brand new. And then half the DNA is the old DNA, and then half the DNA is new. You can see that on the other picture too. So I used the same colors. This blue was the original, the old DNA, and then the red is the new. So you can see that these will be the strands at the end here. Each of these will be the old strand with the new strand, old strand with the new strand. So semi-conservative is the vocab word for that. All right, that's it. I will post some other things on Canvas, some other videos and worksheets for you to do with this too.